So students, we have learned in the previous session about the DNA double helical structure, and also we have learned the double stranded structure of the DNA, how to draw the double stranded structure of the DNA. So that is very important because in the examination they may ask you to draw the double stranded structure of the DNA or even the double helical structure. See, double helical structure is the model that was given by Watson and Crick. So if they ask you to draw the double-stranded DNA or double-stranded polynucleotide chain, you need to draw the way I'm going to draw now. See, it's a very simple diagram, no need to worry much. See, you have to draw some 5 or 6 arrows like this, okay, in one direction, and similarly in the opposite direction also, as much as you have drawn on the upper line there. See, this is going to become, these arrows are going to become what? The pentose sugars or the deoxyribose sugars here. And then you will identify the first carbon atom, third carbon atom and the fifth carbon atom, right? See, this is the first carbon atom of each and every. Let me completely draw all these what is this, nitrogen bases, sorry, pentose sugars on both sides. So these are all the nitrogen bases drawn on both sides now. This is the first carbon atom. These are the first carbon atoms for these pentose sugars, and for these pentose sugars, these are the first carbon atoms. What will be attached at the first carbon atom? That is nitrogen base with the help of which bond? N glycosidic bond. Yes. So nitrogen base attached with the help of N glycosidic bond. Maybe it can be double ringed or it may be also single ringed. See, if one is double ringed, if one is single ringed, the other should be double ringed. So never single ringed, never double ringed, two double ringed or two single ringed nitrogen bases come opposite to each other, because you know already the complementarity base pairing rule given by Erwin Chargaff that we have already seen in the previous session. Okay, so you know the AGCT rule. You also know that complementarity base pairing says that A always pairs with T with a double hydrogen bond, and C always pairs with C. C always pairs with G with a triple hydrogen bond. Okay, so let us consider this as A. Okay, and this one as, see, AGCT, you know, adenine and guanine are what? They are double ringed structures, so this should not be this one. So adenine and guanine, adenine, guanine, adenine. So thymine and cytosine are what? They are the pyrimidines. Purines are adenine and guanine, and cytosine and thymine are pyrimidines. Always purines come or pair with pyrimidines with a hydrogen bond. So if A is there, this should be T, and how many? 2 hydrogen bonds. If G is there, this should be C. How many? Triple hydrogen bond. If A is there, T should come here, double hydrogen bond. A and T, double hydrogen bond. G and C, triple hydrogen bond. So like this, we find different number of hydrogen bonds while base pairs are being formed. Now, identify the fifth carbon atom. If this is the first carbon atom, this is going to be the fifth carbon atom here, right? Fifth carbon atom comes here, and to the fifth carbon atom of every the pentose sugar, the phosphate group will be attached. To the fifth carbon atom of every pentose sugar, the phosphate group is attached. Okay, on both the strands. This is one strand. The other strand also has got the phosphate groups attached to their fifth carbon atom. right? Now see, these are individual nucleotides now. This is one nucleotide, this is one nucleotide, this is one nucleotide. See, in order to form a chain of nucleotides, right, or a polynucleotide chain, there is going to form. See, this phosphate group is attached to the fifth carbon atom with the help of phosphoester bond, and phosphodiester bond is going to be formed while forming the polynucleotide chain. And how is that formed? See, the phosphate group of the next nucleotide is going to be forming a bond with the third carbon atom of this nucleotide now. See, like this. So this is going to be forming the what? Phosphodiester bond, and here OH group is free at a 3' end. So that is why 3' OH, and here the phosphate group is free at this end. This is the 5' phosphate group. Similarly, here also the phosphodiester bonds are being formed, right? And now this is going to be the 5' end, right? Because the phosphate group is present at the fifth carbon atom, and at the third carbon atom the OH group is free. That is why this is the 3' end. So you are seeing the opposite direction. This is 5' to 3'. This is 5' to 3'. They are parallel but opposite in direction. This is what is called as anti-parallelity. Okay, so you have come across one statement while studying about the characteristics of the double helical structure of the DNA. There are two strands which are anti-parallel to each other. This is the meaning of that. So the 5' and 3' end, how the bonding forms, how the N-glycosidic bond is formed, and how the phosphoester and phosphodiester bonds are formed, and how the hydrogen bonds are being formed between the nitrogen bases to form base pair. See, two bases joined together with the hydrogen bonds to form one base pair. This is one base pair, base pair, base pair. This is one base pair and base pair, right? So we measure the length of the DNA based on how many base pairs are there or how many nucleotides are there, right? So nucleotides, see, if there is one nucleotide, that means it will be corresponding with one base pair. If you consider this as one nucleotide, it corresponds with how many base pairs? One base pair. That is why, so to measure or to code the length of the DNA present in the cell of any living organism, we take into account of the base pairs or nucleotides, how many base pairs are there or how many nucleotides are there as the unit of measurement of the DNA. Right? So this is how you need to draw the double stranded structure of polynucleotide, double stranded polynucleotide chain. Double stranded polynucleotide chain. And based on the X-ray crystallography done by Maurice Wilkins and his colleagues, right? The DNA double helical structure of the model was given by Watson and Crick, and later Erwin Chargaff rule also was one of the basis to propose that model. So all that story we have already considered in the previous session. Now in today's session, when the DNA has to be placed inside the cell, what is the length of the DNA? Whether the length of the DNA for all the living organisms, is it the same? Is it same for prokaryotic cell and eukaryotic cell? Is it same for all the mammals? Is it same for all the reptiles? Is it same for all the plants? Is that so? Or is there difference? Or is it correlated with the complexity of the body of the living organism? Yes, partially yes, but may not be true always. Okay, see, maybe the DNA of the simplest organism, the DNA or the genetic material of a very simple organism may be very complex, but the genetic material of a very complex organism may be comparatively simpler, right? So that is why it may not be true that the complexity of the genetic material and complexity of the organism or organismic complexity may not be correlated always. Right? So let us see certain instances, a few instances to consider or to say how the DNA, the length of the DNA is measured in different organisms, and let us consider a few examples. Bacteriophage known as Cy-X174. Cy-X174 has got 5,386 nucleotides. nucleotides or base pairs. Base pair will be mentioned as BP. Okay, so this is the length of the DNA present in this particular bacteriophage. X174, this is a bacteriophage. Bacteriophage, right? And similarly, in other organisms such as lambda bacteriophage, Lambda bacteriophage also has got a number of nucleotides which is far higher than compared to this one, X174. This has got 48,502 base pairs. Okay, so that is in one cell, so many nucleotides are there. And when we look at the length of the DNA in E. coli, that is Escherichia coli, in this organism, which is the bacterium, in this particular bacteria, how much? 4.6 into 10 raised to 6 base pairs. Okay, 4.6 into 10 raised to 6 base pairs. This is the length of the DNA in E. coli. And in human beings, in the human DNA, that is a haploid human DNA. You know haploid and diploid already you have learnt in the previous year, that is in the cell chapter. The haploid content of the human DNA will be 3.3 into 10 raised to 9 base pair. 3.3 into 10 raised to 9 base pair long, right? So these are some of the examples to quote the length of the DNA in different organisms, right? See, when you consider just one set of the DNA in human beings is 3.3 into 10 raised to 9 base pairs, which accounts approximately, see, you already have learned from the DNA double helix model, the distance between each base pair. In one term, there will be approximately 10 base pairs. You know, right? See, in this diagram, when you consider so this is the DNA double helix, helical structure. See, base pairs per each turn will be approximately 10 base pairs, right? The distance between each base pair will be 0.3 nanometer. Yes. So, because the length of each pitch will be 3.0 nanometer, yes or no? So 3.0 divided by 10 will be 0.3 nanometer. Now you may calculate the length of the DNA, that is the haploid content of the DNA when it is 3.3 into 10 raised to 9 base pair. Then you can multiply with 0.3. Then it comes out to be approximately around 2.2 meter per cell. 2.2 meter of the DNA per cell, that is haploid content. Okay, see, that means so you will be having very lengthy DNA compactly placed in a very tiny cell. That cell, the nucleus of the cell will measure up to 10 raised to -6 micrometer. Okay, or you can say 10 raised to -6, what is that? Nanometer. Okay. So if that is the size of the nucleus, and in such a tiny nucleus, how to place this very lengthy DNA? In order to keep the DNA very compactly inside the nucleus, in a very small nucleus, this long DNA has to be folded appropriately, has to be packaged appropriately. How to pack this? How to fold this? Is with the help of certain proteins. Okay, see, this packaging of the DNA is different for eukaryotes and for prokaryotes. See, it depends upon the complexity of the DNA, or depends upon how lengthy the DNA is. Based on that, the complexity of the packaging also will be there in different cells. Okay, so we are going to study the packaging of the DNA for prokaryotes and eukaryotes now. So, packaging of DNA helix. Packaging of DNA helix. I just told you why there is a need for packaging this helix. See, the approximate length of the DNA, the haploid content of the DNA in the human cell, in the nucleus of the human cell will be, that is 2.2 meters, whereas the size of the nucleus will be 10 raised to minus 6 meter, not nanometer. I mentioned it wrongly then. So 10 raised to minus 6 meter will be approximate size of the nucleus. How to place such a long DNA in a very tiny nucleus is the question now, right? In prokaryotes, see, in prokaryotes, the DNA will be less lengthier than compared to the eukaryotes. You know, prokaryotic cells will be having a nucleoid instead of the nucleus, right? Or a primitive nucleus. Nucleus will be absent there. So, absence of the nucleus in the sense what? The genetic material is not covered by a nuclear envelope. Yes. But though there is no nuclear envelope in the bacterial cell, there are certain proteins which are positively charged. You know, the DNA is a negatively charged particle, negatively charged molecule. That DNA which is negatively charged, that will be held together, or it will be surrounded by what? Positively charged proteins. But these proteins present in the case of the prokaryotes are very basic proteins, and they are completely different from those proteins which are present or associated with the DNA in the eukaryotes. Okay, illiruvantaha proteins enide, they are completely different from the proteins present in the eukaryotic along with the eukaryotic DNA, right? Here, as the animal or the organism itself is very simple, the complexity of the prokaryotic cells or prokaryotic organism is very simple, and that is why the length of the DNA is also very small, and the complexity of packaging is also not too high. Okay, simple packaging will be there with the help of certain proteins, positively charged proteins. But in the case of eukaryotes, okay, see, prokaryotes simple packaging will be there, but in the case of eukaryotes, of course, the packaging will be helped by certain proteins, and those proteins with which the eukaryotic DNA is associated, those proteins are called as histone proteins. Histones. See, histones are a kind of basic proteins, basic and positively charged proteins made up of amino acid residues. Why did they become the basic proteins? Because the amino acids with which those proteins are made up of, those amino acid residues are basic amino acid residues. The amino acid residues are lysine and arginine. Lysine and arginine. See, remember, this can be a very important 1 mark question for you. So mention the two proteins or two amino acid residues which make the histones. Okay, or they may also ask you the question in some other way, like give the reason for histones being basic proteins. The reason is they are made up of the basic amino acid residues called lysine and arginine, right? So these make a family of proteins called as histone proteins, and histone proteins, there are 5 different types of histone proteins, such as H1, H2A, H2B, H3 and H4. See, there are 5 different types of histone proteins: H1, H2A, H2B, H3 and H4. See, all these proteins, they actually, excluding the H1, just excluding the H1, H2A, H2B, H3 and H4, they form a core of 4 of these proteins. ಈ 4 ಪ್ರೋಟೀನ್ಗಳೇನಿದೆ ಆ 4 ದೇ ಕಮ್ ಡಬಲ್. ದಟ್ ಮೀನ್ಸ್ 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4, they come together and they form something called as histone core. Histone core anta karitare, or it's also called as histone octamer, popularly known as histone octamer. Why octamer? Because 4 proteins, okay, 2 molecules of each, they come together to form one bundle or one core. That is what is called as histone core or histone octamer core anta kuda karibahudu, right? And where is the H1? H1 will not be included in that octamer. H1 will be placed outside that core. This is H1, okay? H2A, H2B, H3 and H4 are here. See, you know, the DNA will be just like the thread-like structure like this, right? So double helical structure we have studied, right? So it is a right-handedly coiled DNA that we know, and it will form highly coiled structure, right? A network-like structure, very thickly associated structure, and that high coiling will be possible only because of the association of the histone octamer and the H1 along with the DNA. How is that? The DNA will be wrapped around it. It will coil around it, or around the octamer like this. Okay? And so this also will be coiling around with the H1. Okay? See, this entire structure along with the histone octamer and H1 and DNA. See, this complex will be called as nucleosome. A nucleosome complex. Don't get confused. Histone octamer means it is the complex of only these 4 proteins. They come together, 2 molecules of each of them, and that forms the core of the histones, and that core of the histones will be called as histone octamer. And H1 will be outside that core or octamer, and see, the DNA, around 200 base pair long DNA. The length of the DNA that wraps around one histone octamer and H1 will be 200 base pair. So DNA, octamer and H1 all together, it is going to form what is called as nucleosome complex or nucleosome model. Okay, see, why is this necessary? Because see, the packaging takes place with the help of these proteins. See, just to convince you people with this, the same diagram in certain books they will give you like this. So this is the histone octamer consisting of H2A, H2B and H3 and H4. Okay, and each of these molecules, they come double, right? So H2A, H2B, H3 and H4, and this is H1. Why is H1 outside the octamer? H1 will act as the entry or exit point for the DNA. Why is that? I will tell you now. See, so the amount of the DNA that is wrapped around, idur sutta sutkondiruvantaha DNA length estirutte anta helidre, that will be around 200 base pairs. You know that the length of the DNA is measured in terms of the number of nucleotides or base pairs, right? So the length of the DNA that wraps around one nucleosome will be 200 base pair long. So this is how a nucleosome is formed. See, this can be an important 2 marks question for diagram, or they may also ask you to draw and explain how the nucleosome model is formed. So you need to mention H1, H2A, H3, H4, everything. So that may be asked for 4 or 5 marks. Remember, this can be a very important question. But the packaging is not yet over. See, when packaging is done, see, the DNA now looks like beads on string-like structure. See, this is one nucleosome, this is one nucleosome, this is one nucleosome. There are many nucleosomes, okay, on a string of DNA like this, and now it appears as if it is a beads on string like structure. beads on string-like structure. Okay, see here, when many nucleosomes are formed on a stretch of the DNA, when many nucleosomes are formed, the octamer of one nucleosome should not be bound or should not collide with the octamer of the other nucleosome. And also that is why, to protect one octamer by colliding with the other, H1 will act as the entry and exit point for each nucleosome for the DNA. Okay, so in the DNA exit ಆಗ್ತಾ ಇದೆ, and this can be an entry point for the next one. Okay, this DNA will go, and once again it will wrap around another octamer like this. Okay, it will wrap around another octamer like this, and one more H1 will be there, right, like this. So it will act as the entry and exit point, and also gives protection to the proteins present in the octamer. Which one? H1, right? So when beads on string-like structure is formed, see, nucleosome is there, and because of this nucleosome, the DNA length has been decreased, right? Because of the wrapping of the DNA around the nucleosome complex, now the length has been decreased. So what is that? Coiling has been done. Now the DNA appears the little thick or coiled structure, right? And this is called as beads on string-like structure. But in between here, see, this is one nucleosome. Okay, this is one nucleosome. So this will be consisting of all histone octamer and also the which one? H1. Okay, in between two nucleosomes there are also certain proteins, and they are also associated with the DNA, because this much packaging or this much coiling of the DNA will not be sufficient to wrap that DNA and pack that in a very compact space, that of the nucleus, right? So in order to pack it even more further, what is done here? There are certain proteins in between two nucleosomes. Okay, and those proteins which are present in between two nucleosomes and such proteins which are not histones, all those proteins are together called as non-histone protein complexes. Non-histone protein complex. Okay, in short, it can be called as NHC complex. It can be called as NHC complex. What is that NHC complex? Those proteins which are present in between two successive nucleosomes, they are called as NHC complex or non-histone protein complexes, right? So here the DNA that is present, see this DNA, see, you know, 200 base pair long DNA is already wrapped around one nucleosome, and there is a small stretch of the DNA between one nucleosome and the other nucleosome, and that DNA is associated with certain proteins called as non-histone proteins, and this DNA which links from one nucleosome to another nucleosome will be called as linker DNA. Remember, the DNA that links one nucleosome to the other nucleosome, or the DNA present in between two successive nucleosomes, will be called as linker DNA. It is the same linker DNA which will be associated with the NHC, which is non-histone protein complex, right? So this is about the packaging, and when entire packaging is done, what happens there? The coiling of the DNA increases enormously. The DNA is enormously coiled because of these two proteins, basically because of the histones and also non-histone proteins. And when coiling is done, so if you observe under the electron microscope, you will see differently colored DNA, and especially when you are going to use the nuclear stains. So when you use the nuclear stain, basically it is called as acetocarmine or acetooricin. You are going to learn that in the practical sessions, right? So the DNA will appear pinkish colored, but some part of the DNA is darkly stained, some part of the DNA is lightly stained. This colorful appearance of the DNA, that is what is called as, that complex of different proteins forming along with the DNA will be called as chromatin. The coiled structure of the DNA which appears colorful under the electron microscope when stained with the appropriate stains. So this is what is called as chromatin. Okay, so you know chromatin means it is the association of the DNA associated with certain proteins such as histones and non-histones, and that appears colorful when it is stained with the proper nuclear stain under the electron microscope. Okay, that chroma stands for what? Color. Chromatin means what? It is something which will be seen colorful, right, which can be observed colorful. That is chromatin. And in this chromatin, as I told you, some part of this chromatin means it is just the coiled structure of the DNA. Some part of that may appear dark compared to the other portion, right? That part which appears very much darker, that will be called as heterochromatin. Okay, heterochromatin. And the part which appears comparatively lighter in color, that will be called as euchromatin. Euchromatin and heterochromatin. You need to understand the difference between the euchromatin and heterochromatin now. Let us consider the euchromatin and heterochromatin. What is that? What is the basic difference between those two? See, euchromatin, it stains lightly. Heterochromatin, it stains darker. Okay, the first difference is stains lightly, okay, and this one stains darker. This is the first difference. Second thing is that, see, why does this stains lighter, why does this stains very much darker? Is because here the nucleotides are not repetitive. That means there is a particular nucleotide carrying one particular nitrogen base, it can be, or the phosphate group. Of course, phosphate group will be there on every phosphate sugar, pentose sugar. So a nucleotide chain, if there is a nucleotide chain of a particular sequence, and if that is carrying some information, if the same nucleotide is repeated many times, then such a sequence of the DNA will be called as repetitive DNA sequence. Okay, so this is not consisting of repetitive sequences, no repetitive sequences. Okay, it consists of repetitive DNA sequences. Repetitive sequences, and because it consists of the repetitive DNA sequences, it can be observed very darkly. It appears very darkly, and also not only that, transcriptionally this is inactive. Because of the repetitive DNA sequences, this is transcriptionally inactive. Transcriptionally inactive. This is transcriptionally active. This is transcriptionally active, right? So these are the basic difference between the heterochromatin and euchromatin. There are the two different types of chromatin. You should know what is chromatin. A definition of the chromatin can be asked, and for 2 marks they may ask you to differentiate between the euchromatin and heterochromatin, right? So this is about the packaging of the DNA, and this is how the DNA can be placed in a very tiny space within the nucleus. See, when I say transcriptionally active and inactive, see, for DNA molecule, there is a particular pathway of expression. That means the information that is stored in the coded language in the DNA molecule has to be transcribed on RNA molecule, and from RNA it can be converted into protein. And this pathway can be represented like this. See, this is DNA, which has the capacity to undergo self-replication. Yes, self-replication, you know from the cell cycle. In the cell cycle chapter you have learnt DNA during the S phase of cell cycle. Try to recollect it. If you don't remember, go back and read once again. Okay, so replication takes place, and the process and mechanism of replication you are going to learn in this chapter, later in this chapter. So that replicated DNA will be transcribed onto nRNA, that is RNA, right? That means the information present on the DNA will be copied onto the RNA, and that will also cause the biosynthesis of RNA molecule. The synthesis of RNA molecule can be done by copying the information from DNA, and the information present on the RNA now will be expressed as protein. It becomes, or it produces proteins by bringing appropriate amino acids in an appropriate sequence. That is what is called protein. Okay, see, this pathway that we study, that is the expressible pathway of the DNA, this is called as central dogma of molecular biology. This is called as central dogma of molecular biology. This can be asked for 1 or 2 more questions, and it is a very simple question but important. Okay, so remember and write it in the simple and similar way like this. Okay, so in the next session we are going to see the further aspects of this chapter, that is the early experiments in this regard. Different scientists, they carried out various experiments while finding the genetic material DNA.